So, as an aside, why do we care about cell spiking in the first place? Well, an answer is that a spike, an action potential generated by a neuron, is arguably the unit of information in the brain. And as an example of this, let's consider what's called the Halle-Berry neuron. So this is a neuron in a human subject that responds to the concept of Halle-Berry by spiking. So a neuron that responds to Halle-Berry by spiking. And here's an example of this. So what's being shown in this figure, this was a publication in Nature in 2005, are different pictures of Halle-Berry. And if you look closely here, you can also look at the reading this week. You could see pictures of Halle-Berry, just photographs of her as a person, a sketch of her maybe, her dressed up as Catwoman or her name. And then what's also being shown in this figure are spikes generated by a single neuron in a human subject. So this is a patient with epilepsy. Electrodes have been placed inside of this patient's brain, and individual neurons from this patient can be observed a lot in vivo. So while this patient is alive, looking at, or conscious, and looking at these pictures. So what I'd like to draw your attention to are these blue tick marks. These indicate spikes generated by this neuron across different trials. So these blue tick marks indicate spikes. And we can see that this neuron is rather responsive. These are different trials getting shown, for example, this image 22 in the first trial is this first line of spikes, and another trial is the second line, and another trial is the third line. So essentially, the more blue tick marks we see here, the more active or spiking this neuron becomes. And we'll talk about spikes as we go along. So notice that this neuron responds rapidly to these pictures, or it responds by spiking to these pictures or concepts of Halle-Berry, but it doesn't respond to other pictures. If we look closely here, like pictures of Bill Clinton or Mark Hamill from Star Wars, I think that's Mother Teresa at the bottom. So this neuron responds actively, or very aggressively. It spikes a lot when it sees pictures or concepts of Halle-Berry. Another question is, what is this picture at the bottom? We'll get there. So it's another way of characterizing spike train activity, and we'll get there at the end of this week. So again, why do we care about cell spiking? Well, spike is arguably the unit of information in the brain. We saw an example of a neuron that spikes in response to this information that Halle-Berry, the concept of Halle-Berry is present, whether it's a picture of her, a drawing of her, or her name. So if we can understand something about spiking in the brain, perhaps we can understand how each neuron responds to the world, and then how the brain processes information. So it's this idea of going from the things we observe, the spike trains activity, to understanding how the brain processes information. If we can understand spikes, maybe we can understand this information processing in the brain. Now, our first step to this understanding is to characterize the data we actually observe. By understanding these spike trains, perhaps we can understand more about information processing in the brain. So we're going to study spiking, at least to start, in this rather abstract way, where we've taken the spike train data we've observed in a neuron, and we've represented it either as a series of tick marks in the top row, or as a series of zeros and ones, where these indicate when a spike occurs in time. This is sometimes called a spike train, which represents a series of discrete events. Now, as another example, we could think about the arrival times of the T at BU Central. We could stand out there and count the times when the train actually arrives. So we take out our notebook and we write down that a train occurs at 12:45, maybe another train at 1:07, another train at 1:15, and so on. And we can simply indicate each arrival time with a tick mark. So as time goes on, when a train arrives, we put a tick mark at a particular point in time. And again, we have an example of this spike train, where here the spike trains represent arrivals of the T. For the most part in this class, we're going to think about spike trains as representing action potentials generated by a neuron. Now compare this series of discrete events when the train arrives to something like computing the temperature at BU Central. Now this temperature wiggles around. There are no discrete events, and we can record the temperature at each moment in time. So there's a distinction I'd like to make there between these discrete events, the arrivals of a train at the T, versus this continuous temperature fluctuations we could observe at each moment in time. Right now, we're focused on these series of discrete events, just like we have for a spike train. A series of discrete events, they could be arrival times of the train at the T, or these discrete events could be when spikes occur, produced by an individual neuron. So that's our focus now. So returning to the experiment, our collaborator does not provide us just one example of voltage activity over time. He provides, in this case, multiple trials of data. So for example, here's trial 1. A stimulus is delivered. Maybe it's some chemicals applied to the bath, and the spiking activity is recorded as a function of time. We're showing the spike train data. So that's trial 1. This experiment is then repeated. The same stimulus is delivered. We record another spike train. This is repeated in trial 3, and so on, all the way up to trial K. So in these series of notes, we'll use these red symbols to indicate the stimulus delivery, and then we have our responding spikes. So this is the spike train in response to this stimulus. So one thing to note here is that although the same experiment is applied each time, the same stimulus is applied at the beginning of each trial, the results are different. We get different spike trains that appear in trial 1, trial 2, trial 3, and so on. So there's some randomness we don't control. There's some randomness that occurs in these data that's not related to the presentation of the same stimulus over and over again. Same stimulus applied, we get different results. So what could this be? What could be causing this randomness? It could be lots of different things. It could be a changing chemical environment that's happening in the slice. It could be other inputs coming into the neuron beyond the stimulus. You could probably think of many other things as well. It's the parts of the experiment that we don't control can give rise to this randomness. Okay, so how do we characterize these data? We've now collected multiple spike trains across, let's say, K trials. So we have all of this data. How do we characterize it? There are many answers. We can start in a simple way, which is always a good way to start, visual inspection. And maybe we perform a verbal model of these data, and we can say after the stimulus is applied, the neuron spikes. That's a fine way to start. So we observe something about the data. But we'd like to get more quantitative than this and start characterizing the data using quantitative methods. And we're going to discuss 5 measures here. We'll compute the spike number histogram, what's called the firing rate. We'll look at the interspike interval histogram, the rastogram, and the post-stimulus time histogram. So these are 5 measures we're going to summarize here in this lecture and on Wednesday, and then we'll implement in MATLAB on Friday. So again, we'll describe these tools first. You'll see some examples, and then we're going to apply them and actually use them in practice. Okay, so to start, let's compute something very simple. Let's just count the number of spikes in a trial. For example, we can count the number of spikes that occur in trial 1. So here's trial 1. We have our stimulus delivered at the beginning of the trial, shown here in red, and then we have a certain number of spikes that appear over time. In this case, we can count up those spikes, and I get 15 in this case. 15 spikes occur in the first trial. And we're going to represent that with a symbol. N sub 1 will set equal to 15. Where the notation in this case, N, we can think of as representing the number of spikes. And this is occurring in trial 1, so that subscript indicates trial 1. And we can repeat this procedure for different trials. So let's count the number of spikes that occur in trial 2. Here's trial 2. We can count up the number of spikes. The symbol we'll use in this case is N sub 2 to represent the number of spikes, N, the number of spikes, in trial 2. And we find in this case that there are 13 spikes that occur in trial 2. So if we repeat this procedure for each trial, we generate a long list of numbers, which represents the number of spikes in each trial. We saw that in the first trial, N sub 1, we have 15 spikes. In the second trial, N sub 2, we have 13 spikes. And so on. We can repeat this procedure until we get up to the kth trial. N sub k in this case is also equal to 15 spikes. So we repeat this procedure for all trials, trials 1, 2, 3, all the way up to trial k. So one thing to note, there are k total numbers in this list. We're generating this list of numbers, the number of spikes in each trial. There are k total numbers in this list. That's the number of trials that we observe. Each trial has a certain number of spikes, which we're recording here. Another thing to notice is that we've compressed the data again. We've gone from our spike trains, which tell us when in time the spikes occur, to this list of numbers, the number of spikes in each trial. So we've compressed the data. So in doing so, when we've done this, what is lost? Right? All we have left here, let's say for trial 1, is the number of spikes that occur in that trial. So what have we lost? Well, we don't know when those spikes occurred in that trial. Did they occur right after the stimulus? Did they occur sometime later after the stimulus? Were those spikes evenly distributed throughout the trial? Or were they bunched up somewhere? We don't know that. All we know is that 15 spikes occurred in trial 1. So we've lost that information about the exact timing of the spikes. All we know is the total number of spikes that occurred in each trial. Okay, so that's fine. We've compressed our data a bit more. We're just representing the number of spikes in a trial. Now what? What can we do with this list of numbers, the number of spikes in each trial? What can we do with that list? So here it is, the list of numbers. Number of spikes in trial 1, trial 2, all the way up to trial K. We have this list. As an answer, there are many things we can do with this list of number, this vector. We've thought about ways to manipulate and visualize vectors in MATLAB. So we have maybe some ideas. We're going to think about two categories for what we can do for this list of numbers. The first is we can visualize it. Let's have a look at it. Always a good place to start. And the second week is we can start characterizing it and computing some interesting quantities from these data, or different types of statistics. Okay, so let's start by visualizing this list of spike counts. So one method we could use to visualize the list of spike counts is we can write down all the values and look at them. So here's our vector. We've printed it out at the command line, let's say, in MATLAB. This is our long vector. And what does this first number represent in our vector? Well, remember, this first number represents the number of spikes that occur in trial 1. So this is N1. What does that second number represent? Well, that's the number of spikes in trial 2, and so on. So what we're writing out here is the number of spikes that occur in each trial. And we're just printing it all out. And we can ask questions about this list, like what types of values do you see? And if I stare at this list, I see values that maybe around 10, 14, 11. Seems like there are a lot of those values. Every once in a while, larger values like 22. And maybe the smallest value I see, just staring at it briefly, is 5 here. So we get a sense for the range of values, the number of spikes that occur across trials. And we can ask another question, like if you were to pick a trial at random from the experiment, how many spikes do you expect to encounter? And if you're interested, I'll set up learning catalytics so you can go and answer this question. If you were to pick a trial at random, how many spikes would you expect to see? There's a question we can consider. Just by looking at the data in this way, by plotting out the data and looking at, or printing out the data in this case and looking at it. Another question, how many trials are there? How can we answer this question, the number of trials? Well, we know that the first number is the number of spikes in trial 1, second number is the number of spikes in trial 2, so 12 must represent the number of spikes in trial 3, 7 is the number of spikes in trial 4, and so on. And if we keep counting up all these numbers, we'll eventually figure out the number of trials. So it's the length or the size of this vector will tell us the total number of trials. And if we were very patient, maybe we could count up all of these numbers by hand, and you might think about how we would compute the size of this list in MATLAB to determine the number of trials. And it turns out the answer in this case is 200. So there are 200 numbers. If you're very patient, you count them all up, you should find that there are 200 numbers. This is not something we would do by hand. This is something we would ask a computer to do. Okay, so that's one way to visualize the list of spike counts, is we can print them out and look at them. We can also plot the values and look at them. We'll do this in MATLAB on Friday, but just to get us started, here's a plot of the number of spikes on the vertical axis versus the trial number on the horizontal axis. And we can ask similar types of questions, like what types of values do you see? And we see in this case that there are values that maybe go down to 5 and all the way up to 20. And if we were to pick a trial at random from the experiment, so out of all 200 trials, pull one out at random, how many spikes would you expect to find? Well, a reasonable guess is we would expect to find something maybe in the range of 15 to 10, maybe a little bit lower or a little bit higher. Somewhere in this range. We wouldn't be surprised if we pulled out a trial at random and got 12 spikes. We'd be very surprised, however, if we pulled out a trial at random and saw 100 spikes, since that doesn't seem to appear in our data at all.